All right, good morning. All right, good morning. Now let's get started. I think we stopped here, right? Last time. So we're talking about the transition between the different macronutrients. We know that fat cannot be converted back to the protein or carbohydrate, but both carbohydrate and protein can all be able to convert it to fat if they have more than enough, right? And our body need. So, and we also ask question, what our body going to do with the extra fat. So you guys know, just want to get rid of, right, by burning fat. So, and you also know that the most important pathway for burning fat is fatty acid oxidation, right? So, and about obesity, I guess everybody knows, right? The main cause is the imbalance between food intake and energy expenditure, right? We either eat too much, or we don't exercise, or have a bad kind of habit, cannot burn enough energy or something like this, or imbalance, right? And other thing is important than the rate of obesity, right? It's just keep going up. And the United States and also leading the rate of obesity. So the number one, the high rate of obesity, right, in the world. So that's really problematic. And if you look at this, similarly, the obesity rate in the United States is about 33.8%. So like 3 people, you have one, right, obese. And it's just the map of obesity rate. And the light blue that indicated obesity rate less than 10%, and different colors indicate different rate of obesity, and red, dark red, right, is like more than 30%. So if you look at the history, like 1996, well, actually pretty good, right? We kind of have a obesity rate less than 20%, but 2009, you see lots of states and become red, orange, or red, right? And if you look at this slide, even worse, like 2015, I guess more than half, maybe 2/3 of states in the USA, and become red. So and some states even become purple. Purple color indicated obesity rate is higher than 35%. So it's really problematic. So usually we use the body mass index to indicate the obese degree or something like this, right? So BMI 20 to 25 is kind of ideal, the desirable range. So we wanted to keep our BMI within the 25. Okay, so 20 to 30, between 25 to 30, that we consider the overweight, and 30 to 40 is really obesity, and greater than 40 is severe obesity. And this just tells us the gender difference in the body composition, the percentage of water, percentage of fat, mass. If you compare men to women, and basically men had a more percentage of water, and but this actually the normal composition of body composition comparison, right? So usually women have a more fat, right, storage fat compared to women, compared to men. And not just the total amount of fat in the body in terms of obesity, and more importantly, the distribution of fat in the different part of the body is most important. So based on the distribution of fat, the obesity can categorize two categories. Like one is called abdominal obesity, or the meal pattern or apple obesity. That means most of the fat actually accumulate in the abdominal region, right? So, and this type of obesity more common in men, and so this we call apple or man pattern of obesity. And the other type of obesity is the female pattern or pill pattern. So this female pattern of obesity, the most of the fat accumulate in the in the hip and thigh region, right? And also the different type of obesity, they have a different relationship with a correlation with the metabolic syndrome, or the risk of a metabolic syndrome. For example, the apple obesity or abdominal obesity is more closely related to the development of metabolic syndrome, like cardiovascular disease, hypertension, right, or diabetes, type 2 diabetes. And the female pattern obesity is less correlated with the metabolic syndrome, right? So as I just described in the last slide, this is just more information about fat distribution. Sometimes we also call it upper body fat distribution or visceral obesity, right? Same thing. It's kind of abdominal obesity. Is closely correlated with the metabolic syndrome, and the so we also use the so-called waist to hip ratio to indicate the risk for metabolic syndrome, right? So for example, if the waist to hip ratio is greater than 0.85 for women or greater than 0.95 for women, and in this ratio, that indicated you probably have a high risk for the development of metabolic syndrome, right? So we usually, again, use the waist to hip ratio to indicate the risk for the metabolic syndrome. And the other thing is important is that the so-called ectopic fat deposition. What I mean is, so we know that most of our triglyceride or fat is stored in the adipose tissue, right? So adipose tissue should be the right place for the storage of a fat or triglyceride. So if triglyceride or fat is accumulate or deposit in the non-adipose tissue, for example, liver and muscle, right? And this kind of condition or situation is called ectopic fat distribution. So this is not good. So if someone has a more ectopic fat deposit in the non-adipose tissue, that are closely related to the metabolic syndrome, right? So now let's talk about the adipose tissue function. So adipose tissue has actually different type of adipose tissue in our body, right? Main two types, white and brown adipose tissue. So white adipose tissue and brown adipose tissue, they have a different function. So white adipose tissue, we know mostly is for the storage of fat, right? The triglyceride or fatty acids. And this is the storage of a fat is serving as called fat buffering system, right, in the lipid metabolism. So we wanted to store the most of the fat in the adipose tissue. So don't allow the fat to spread to the other tissue. That is not good, right? So the adipose tissue storage fat is a good thing, to prevent the ectopic fat deposition or prevent the metabolic disease. So if some reason that adipose tissue cannot store fat, there is a disease like leptin deficiency. The patient almost has no subcutaneous fat, right? The fat stored under the skin, they almost no subcutaneous fat. And most of fat actually accumulate in the abdominal region, like liver and muscle, right? Have lots of fat, stimulate the non-adipose tissue. So this type of patient have a severe type 2 diabetes. So again, adipose tissue storage, the fat storage is a good thing, right? It's a buffering fat system to prevent the lipid dysregulation. And second function of white adipose tissue is the glucose homeostasis. If you still remember, the adipose tissue and muscle both are the main tissue that express GLUT4, right, and uptake glucose into the tissue. So the tissue is, this adipose tissue is also important to uptake glucose from circulation, right, and in response to insulin stimulation. So it also contributed to glucose homeostasis. So that means can reduce blood glucose level. Okay, so the third important function of adipose tissue is endocrine function. So not fat cell or fat tissue, not just stored triglyceride fat, but very importantly, secreted important hormones, right, and regulate the whole body metabolism. So adipocyte, that this is just a cartoon show the adipocyte. So adipocyte is very different from the other type of cells. Not just because the size is bigger, much bigger, but the whole cytoplasm filled with the lipids, right? The whole lipid droplets, and the nucleus, because of so much lipids in the cell, so they push the nucleus to the edge of the cell, right? So have a very little cytoplasm. So this is the real adipocyte, right? So this is something that I did when I did my postdoc at the NIH. So I isolated a fat cell from the rat adipose tissue and looked at under the microscope. So you see this is not a bubble, right? It's actually adipocyte, the real adipocyte. So you see the, filled with the oil lipids. You almost cannot see the nucleus. So adipocyte we know has a insulin receptor, right? so insulin responsive cells, and also has a group 4. So insulin stimulates group 4 translocation to the cell surface and uptake glucose, right? This we already learned. So it's very important for the glucose homeostasis, right? By uptaking glucose from the circulation, we reduce the blood glucose level. And also adipose, the adipocyte is important for fatty acids and triglyceride synthesis, right? Particularly triglyceride synthesis. If you still remember, under the fat condition, right, we absorb a lot of fat or fatty acids from the diet, and fatty acids being transported through the lipoprotein. You remember which lipoprotein actually transported fatty acids or triglyceride from intestine to adipose tissue? Chylomyocrine, right? So before triglyceride being uptake to the cells, triglyceride need to be broken down, right, by LPS to release fatty acids, and fatty acids are uptaken by fat cell and then re-acidify in the adipocyte, right, to the triglyceride and stored in the adipocyte. So the triglyceride synthesis activities are very active in the adipocyte. And of course, express the LPL, right, lipoprotein lipase, the adipocyte, so that can break down the triglyceride to release fatty acids, and fatty acids, and again, uptake by fat cell and re-acidify to triglyceride. And adipocyte also express the receptor, the adrenaline receptor. This receptor actually is response to which hormone do you guys know? Catecholamine, right? Catecholamine. So catecholamine levels increase during the fasting. So catecholamine can bind the adrenaline receptors, right? And to activate which enzyme in the adipose tissue, adipocyte, that can hydrolyze triglyceride, release fatty acids. Hormone-sensitive lipase, right? So hormone-sensitive lipase then can be activated by the adrenal receptor signaling, right? And release fatty acids. So something the insulin signaling pathway, like a top part, is activated during fat condition, right? And the bottom part, the adrenal receptor signaling is activated during fasting, right? So this sort of a summary of a fat cell function in the regulation of lipid metabolism, particularly triglyceride synthesis and triglyceride breakdown, release fatty acid during fasting, right? And the last slide that we talk about is a very well-known and basic function, right, of adipocyte: storage triglyceride and release fatty acids during fasting, something like this. And now I'm going to talk about the secretory function of adipocyte, right? Besides those basic functions, adipocyte can also secrete very important hormones, cytokines, and lipids like fatty acids, right? We know release fatty acids during fasting to provide energy for many other tissues, right? And leptin, which is the first hormone that was discovered in 1994 that released from adipocyte. So leptin is very important hormone in regulation of whole body energy metabolism. And adipocyte can also secrete some cytokines that related to inflammation, right? And some factors called angiotensinogen, and this kind of factor can regulate the blood pressure or something like this, right? And adiponectin is another important hormones that can be secreted by adipocyte and also regulate the energy metabolism and other immune functions, right? And resistin is the third factor that can be secreted by fat cell, but compared to leptin and adiponectin, resistant function is less being studied, right? But of course, besides all these three factors, fat cell can secrete lots of other type of factors, although their function is not known, right? Actually, two of factors that new factors, that released by adipocyte that we kind of interested in. So our lab can study that two fat cell secreted protein, and they do have a very important in regulation of obesity and insulin resistance and diabetes, right? Okay, so now let's talk about the specific two hormones that are secreted by adipocyte. First and most important one is leptin, right? So leptin is secreted from adipose tissue into the circulation, right? And then can go to the two main tissues, brain, right? The two main target tissue, brain and the muscle, right? And regulates the activity in these two tissue organs. So for the first of all, the first function, also called the central function of leptin, is to regulate appetite and energy expenditure, right? So leptin basically can inhibit the appetite and increase energy expenditure. So leptin is actually good hormone, right? Can secrete from fat cell and go to the brain to regulate appetite, food intake, and also increase the energy expenditure. And the peripheral function of leptin. So leptin can also target muscle to induce or to stimulate the fat oxidation in the muscle. Muscle is a very important tissue for fatty acid oxidation, right? We know, if you still remember, 1/3 of fatty acids actually go to the muscle for oxidation, right? So, so muscle plays very important in energy expenditure, to oxidize fatty acids, right? To get rid of fatty acids. So leptin does two things again: central function to inhibit appetite, increase energy expenditure, basically through the increase the muscle fat oxidation, right? Any questions about leptin? Okay, so regulation of leptin production by nutritional status and insulin. So basically when we put animal or human fasting, for example, for 24 hours, and that will cause the 90% reduction in leptin gene expression. So in general, if we are under fasting condition, leptin production will be reduced. And refeeding, so if you put up, for example, mice under fasting for 24 hours, and then you give food back to the animal, refeed them, then leptin can be quickly induced, right? Like just 3 to 6 hours, leptin can be reinduced. And also insulin, if you give the mice, the fasting mice, the insulin can also stimulate leptin expression, right? So, so why does the body make less leptin during fasting? Thinking about, what the leptin does. Why fasting can reduce the leptin? Yeah. Right. Yeah, great. So, fasting, we want to save energy, right? We don't have enough energy, so we wanted to reduce leptin and to save, energy, right? To not allow too much energy being wasted, right? Okay. Okay, interesting thing is OB mice. So, the OB mice basically is a leptin deficiency. The OB mice, they don't have leptin, right? Leptin deficiency. So so people just didn't know before 1994 when leptin was discovered, right? People already noticed that, a specific strain of obese mice. They are very obese and they eat a lot and gain a lot of weight. Like if you compare to normal mice, their body size almost double. But people didn't know what happened to these obese mice, figure out what causes their obese. So a group of scientists, they did the experiment. They actually crossed the circulation, right, between normal and obese mice. So that means that obese mice blood can go to the lean mice, or the normal mice blood can also go to the obese mice. So they kind of cross the circulation. And for a period of time, then eventually they found obese mice and become the body weight actually reduced, the lost body weight. So eventually, so they suspect something in the circulation, right? Does the effect reduce the body weight in obese mice? Some circulating factor that from normal mice, right, go to the obese mice and reduce the obese mice body weight. So in 1994, a group of scientists, I can't remember, in Rockefeller University, I guess, that actually they cloned the obese gene and discovered this obese gene, actually the leptin. So leptin is expressed again, produced by a fat cell, and can release into circulation, regulates the appetite, energy expenditure, fat oxidation also. So then they inject the leptin back to the OB mice. That can also have a significant effect, almost can 100% rescue, right, the OB mice. The body weight return to the normal. So again, leptin can suppress the appetite, right? And okay, so that 1994, the people were so excited, about the discovery of leptin. So every obese people, have a hope, right? We can use leptin to treat obesity. But the problem is people realize that actually obese people or mice, they actually have more leptin in the circulation. So they found that this obese patient or obese mice, they are leptin resistant. right? So they have more leptin, but they have a lower leptin action. So it's more complicated, you know. So okay. So the second type of genetic obese mouse model is the DBDB mice. And DBDB mice also become very obese, right? And they also hyperphagic, that means they eat a lot. And they also become very severe type 2 diabetes. So they have a hyperinsulinemia and hyperglycemia. They have a very high glucose level and also very high insulin level. And they are severe insulin resistance, right? So the problem, and people realize that if they do the cross circulation, didn't help at all. So they realize eventually these mice actually leptin receptor deficient. They don't have a leptin receptor. So although you have a leptin level, right, and even higher, but the leptin cannot function. So definitely if you give a leptin to this DBDB mice, it will not help, right? Because they don't have a receptor. Okay, so that's the leptin. Now let's talk about adiponectin. So adiponectin, later on, you know, people also discovered, fat cell can secrete another hormone called adiponectin, and adiponectin has two different tissue target, liver and muscle. So what adiponectin does to these two tissues is to increase fat oxidation, right? Increase energy expenditure. So adiponectin is also a good thing. And the type 2 diabetes patient, obesity subject, they usually have a lower adiponectin level, right? The adiponectin does not have a central effect. Basically, the peripheral effect, you know, two main tissue target, liver and muscle. And main effect is to increase the fat oxidation, right? Fatty acid oxidation, so can burn more fat. But recently, a lot of studies are shown that adiponectin also has other different important function, like regulates immune response or something. But you just know, but you don't have to remember, right? Okay, so similar to leptin, the adiponectin can also be regulated by fasting. Fasting also decrease adiponectin, right? Similar reason, right? Also, you want to save energy, and refeeding can also rapidly increase adiponectin, and insulin can also stimulate adiponectin expression. And again, I mentioned type 2 diabetes, insulin resistant patient, the plasma leptin level usually decrease, right? Okay, so for summary, that both leptin and adiponectin, the summary, but a little bit more detailed about the mechanism, how both leptin and adiponectin functioning in energy expenditure, right? Increase the fat oxidation. So if you still remember the AMPK, right? AMP, activated protein kinase, right? We talked about this earlier, right? Is the target of leptin and adiponectin. So adiponectin and leptin basically can activate AMPK kinase, and then induce or increase fat oxidation. So we have more steps between these two. These 2 points, right? And we have a slide later to explain, the more detailed pathway or mechanism, how leptin, adiponectin activate AMPK and activate fatty acid oxidation. So increased fat oxidation, of course, we can have an effect by reduce to on the circulating fatty acid triglyceride and insulin resistance, right, being improved. So AMPK is a serum threonine kinase, and it's a fuel sensing enzyme. So AMPK can be activated during the energy crisis, right? Energy, for example, during starvation. We have a low energy in the body that can stimulate AMPK activation. The AMPK is very important. The enzyme can regulate both oxidation of fatty acids and the glucose, right? So once the AMPK is activated, so AMPK does two different things. One is to inhibit ATP consuming anabolic pathway, right, or save ATP. And the other thing is to AMPK does is to stimulate ATP producing catabolic pathway, right? So that will make sense, right? When we have a lower ATP levels, right, then AMPK can be activated and to regulate the two different metabolic pathways. Okay, so this diagram is very important. I hope you guys can remember to memorize some of the pathways, right? So again, mentioned the adiponectin and leptin. So this is very important. It's actually the muscle cells, if you still remember. Both leptin and adiponectin can target muscles, right, and stimulate fat oxidation. So this represents the muscle cells, and on the muscle cell surface there is two receptors, leptin and adiponectin receptors. So both adiponectin and leptin can bind receptor and activate AMPK, right? So AMPK, if you still remember, I hope, then can phosphorylate ACC, right? ACC is a key enzyme for fatty acid synthesis, right? Or for this reaction, the malonyl-CoA formation from acetyl-CoA, right? Remember? So ACC gets phosphorylated. What is its activity? Reduce, right? Reduce the ACC activity. So once ACC gets phosphorylated, ACC activity is reduced. And so what happened to the ACC catalyzed reaction, the formation of malonyl-CoA, will be decreased, right? So once the ACC gets phosphorylated by AMPK, the malonyl-CoA formation reaction will be slowed down or decreased. So you have a lower malonyl-CoA level, right? And this is the mitochondria, right? Beta-cross oxidation occurs in the mitochondria. And malonyl-CoA is a very important regulators of CPT1, right, on the mitochondrial membrane. So CPT1 is the key enzyme that control fatty acids transported from cytosol into the mitochondria for beta oxidation, right? Still remember? So lower malonyl-CoA will cause higher CPT1 activity, right? Is that correct? You still remember that we learned it before, right? So because malonyl-CoA is an inhibitor of a CPT1, right? When we have a lower malonyl-CoA, then we should have a higher CPT1 activity, right? So how you connect the activation of AMPK to fatty acid oxidation? Just simply, when AMPK is activated, what happened to fat oxidation? Increase or decrease? Increase, yes, right? So this is a connection, ACC, melanoid-CoA, and CPT1, right? Okay, so and the other important thing I want to mention, if you still remember, the muscle glucose uptake or GLUT4 translocation by two different mechanisms, right? What is the first important mechanism? What factors, hormones can stimulate GLUT4 translocation from intracellular to cell surface in muscle? Insulin, right? And I think I also mentioned the other important factor that can also, yes, muscle contraction. So muscle contraction during exercise, right? Even you don't have a higher levels of insulin, then GLUT4 can also be translocated to the surface and uptake glucose. So this is the mechanism here, the AMPK. So when we have a higher muscle contraction or during exercise, AMPK can also be activated. So AMPK is the important enzyme and can activate signaling pathway that can promote GLUT4 translocation, right? So the detailed mechanism, the pathway is not very clear, but you just want to remember that AMPK activated by muscle contraction and can stimulate GLUT4 translocation to the cell surface, right? Okay. All right. Let's take a break. All right. So we have a couple of summary slides I'm not going to go through them, and you guys just okay, now let's move on to okay, so metabolic fate of micronutrients in response to food intake and metabolic fuel utilization switch, right? So in this session, we're going to talk about more detailed and how our body changes the metabolism and after meal or during the starvation. So we give us kind of a time period, right? So, for example, we're going to talk about two different phases, physiological state. First is absorptive phase or fat condition. So we define fat condition by like 2 to 3 hours after meal, right? Usually, like we eat breakfast at 8 o'clock, let's say, right? And usually we eat lunch like 4 hours later, 12, as a typically, right? So if we don't eat lunch or delay lunch or skip lunch or whatever, then after the 3 to 4 hours after the meal to the days and this time period, you consider the post-absorptive phase or fasting, right? So if you don't eat lunch at 12, right, or skip lunch, and then you start starvation or start fasting. So and the second thing that we're going to talk about in this session is how does the body switch metabolic fuel during starvation. We have a kind of definition or theory to explain the fuel utilization switch with in metabolism called a Randle cycle, right? And then we also going to talk about how does the body know when to switch metabolic fuels. So regulation about fuel utilization switch. So like hormone, substrate, enzyme transporter, they are important factors that involve the regulation of fuel utilization switch, right? So for example, during the absorptive phase or during fat condition, because we eat, right, so we get dietary carbohydrate, protein, or fat. So our body during the this 4-hour period, we usually be able to use energy that are from diet, right? And after 4 hours, if we skip lunch, then we start fasting, then our body has to do something because we don't have a food dietary sources of energy anymore, right? So we start to use the endogenous energy fuel or stored energy fuels or also make energies, right? Energy fuels. So the switch from, the exogenous or dietary energy source to endogenous source. This is one type of a switch. And the other type of a switch is that during the fat condition, that 4-hour period, we usually mostly use the glucose as energy, right? Use the carbohydrate as energy. But during the fasting, then we start using more fatty acids or ketone bodies for energy. So we also, our body also does a switch from utilizing carbohydrate to utilizing fat or fatty acids as energy. So different type of switch, right? The sources of switch and also the type of energy fuel switch. Okay, so a standard meal, it usually has a carbohydrate, 90 gram of carbohydrate, 30 gram of protein, and 20 gram of fat, right? It's a typical diet, meal. So for carbohydrate, 90 gram glucose in meal, first of all, we absorbed it from the diet, right? And if you remember, the enterocyte is an important cell for absorption. So first step is the SGLT1. This is a glucose transporter, right? Transport the glucose from gut lumen into the enterocyte. And then the second level of the transport is a GLUT2 mediate transport of glucose from enterocyte into the portal vein, right? Circulation. And once glucose enter the circulation, then can stimulate insulin release by beta cell. So I guess you guys all know this, right? And then once the glucose enter the portal vein, the liver takes up 20% of glucose then from the diet. And glucose can be rapidly converted to glucose 6-phosphate by glucokinase, right? Which is a first key enzyme, right, in the glycolysis. So in the liver, so glucose under the glycolysis in the liver, once it's absorbed from the intestine, and most of the glucose under the fat condition. Well, maybe I'll ask you a question, right? So how is glucose metabolized in the liver in the fat condition? You know, once it's uptaken from the portal vein, right, circulation. Glycogen, right? To convert this glycogen under the fat condition. So most of the glucose uptaken from the circulation by the liver being used or converted to glycogen stored in the liver, right? And glucose usually not completely oxidized in the liver, even under the fat condition, right? Usually it does partial oxidation, so produce lactate and save the glucose. During the fasting, of course, the glucose will be also saved, right, for the gluconeogenesis. Okay, so this slide just show us the glycogen synthesis in the liver under the fat condition. If you the percentage of the dietary glucose versus the glucose from gluconeogenesis of amino acids or lactate, you can see the majority of the glucose in the glycogen under the fat condition, right, is from diet, right? So 60 and 70% of dietary glucose are being used for synthesis of glycogen, right? So and 30 to 40% of the glucose in the glycogen in the liver, basically from the lactate and amino acids through the gluconeogenic pathway, right? So for example here, the dietary glucose, 60 to 70%, right, directly converts to glucose 6-phosphate and then glucose 1-phosphate, right, enter the glycogen synthesis pathway. And because of the glucose 6-phosphatase activity is quite low under the fat condition, right? So the reverse reaction will not happen a lot. right? Will not happen under the fat condition. So again, this will be the most this will be the major direction of the glucose metabolism, right? Glucose to the glycogen synthesis pathway is the most important pathway under the fat condition. Again, 60-70% of glucose in the glycogen is from diet. And some of the glucose can also undergo glycolysis, right, and generate lactate. And lactate under the fat condition will not be released, right, but enter the gluconeogenic pathway and to generate glucose 6-phosphate, right, and also then, enter the glycogen synthesis pathway. And another sources of glucose is from amino acids. We also know, right, when we learn amino acid metabolism under fat condition, many amino acids can be enter the gluconeogenic pathway, right, and be converted to glucose and store in the form of glycogen. So this is the pathway that we talked about earlier. So glucogenic amino acids, right, that from diet also, that can be converts to glucose and stored in the glycogen, right? So again, during the fat condition, 60 to 70% of glucose, dietary glucose, being converted to glycogen, right? And the dietary amino acids can also be converted to glucose and stored in the glycogen. And the main reason that we go to this direction because of the glucose 6-phosphatase activity in the liver under fat condition is very low, right? So this will be the main direction of the glucose metabolism in the liver under the fat condition. And 22% of glucose from the diet actually bypass the liver, will not be metabolized in the liver, right? And be directly, it's not. Sorry, I said it wrong. So 22% of the glucose from the diet actually metabolized by the liver, right? And the brain will also take up 22% of dietary glucose for direct oxidation, right? And another 2 to 3% of glucose being metabolized in the other tissue through the glycolysis pathway, right? And most of the glucose being uptaken by muscle and adipose tissue in an insulin-dependent manner. That means through the GLUT4 transporter, right? Activity. So you just get a sense, of how much kind of a proportion, how much glucose metabolized in different tissues, right? Any questions? And muscle actually takes a lot of glucose, like 20 to 25% of glucose. And glucose that being taken up by muscle, 50% goes to the complete oxidation, another 50% go to the glycogen synthesis pathway, right? So adipose tissue, we talk about a lot, right? The glucose being metabolized in the adipose tissue primarily for the synthesis of glycerol backbone, right? Glycerol 3-phosphate, and used for what? Yeah. Triglyceride synthesis. Yes, thank you. Right? Okay, so this is a diagram. Just give you a idea how glucose being distributed into the different tissue and how the glucose metabolize under the fat condition, right? So from the meal, like we have about 90 grams of glucose from the meal, and 50 to 20 grams goes to the brain for energy, and 20 grams go to the liver for glycogen synthesis, and also part of glucose will go to the glycolysis, right, for ATP production. Again, just a partial oxidation, right, to produce ATP. And 20 to 45 grams go to the muscle, and half of them for synthesis of glycogen, and another half for complete oxidation of ATP, and 2 grams go to the edible tissue, right, for making backbone, right, glycerol 3 phosphate, the triglyceride backbone. Okay. All right. So this is the this slide show us the dietary carbohydrate, fate of dietary carbohydrate during absorptive phase. That means 2 to 3 hours after meal. So what happened to the glucose, right? 2 to 3 hours after we eat. So that involves a lot of tissues, right? Almost the whole body tissues. So dietary carbohydrate, once absorbed it, right, into the enterocyte, some of the glucose are used by small intestine. If you still remember, we talked about, right, under fat condition, 80% of energy is from 3 important amino acids, right? And another 20% of energy is from dietary glucose. And 22% of ingested glucose go to the liver and be metabolized, right? The main pathway means the glycogen synthesis, right? But of course, some of the glucose can be used for fatty acid synthesis if we have more than enough glucose, right? And synthesized triglyceride is released as the VLDL. And non-metabolized or non-uptaken glucose will eventually go to circulation, and 22% go to the brain for energy, and 2 to 3% to other tissue. That's the glycolic tissue, right, for energy. And these two tissues